Hello everyone, welcome to my YouTube channel. So we've been talking about factors that could influence IHC staining, okay? That could actually lead to poor IHC staining. I'd already made some points, so I'm going to continue to talk to us on that. So the next point I want to talk about is poor tissue quality. Poor tissue quality. Before we continue, my name is Dr. Emmanuel Obodo. I'm a biomedical scientist and I'm also a lecturer in biomedical science here in the United Kingdom. Right. So when we talk about poor tissue quality, what exactly are we talking about? Obviously, we already know that the basic histopathology means there is a tissue and it has to come in a fixative, okay? And then when ready, then there can be cut up, and of course the tissue processing will now go ahead, okay? Now, what could make tissue to be poor? What could lead to poor tissue quality? Now, when there is poor tissue quality, what it means that there could be maybe autolysis, or maybe there is what necrosis. So autolysis in the sense that there are some biological activities going on in that very tissue that has caused a degradation of that very tissue, okay? So that tissue has deteriorated, and the deterioration of that tissue means that its biological properties has been compromised. And because it's been compromised, it then means that you may not be able to get the required result or the required staining that you would have expected when you do your IHC staining. Now, another thing is that when you talk about necrosis, it means that there is a cell death, okay? So this cell death means that, again, the cell death means that the properties of the cells, what you are looking for, the antigen has denatured perhaps, or maybe the antigen is no longer active. Are you getting me? Maybe even the binding site may have shrunk, as the case may be. So the death of the cells means that the potency of the antigen, of course, has been also compromised, and that way the antibody may not be able to bind effectively to be able to give you a good IHC staining. So that is why one of the main factors that could lead to this very poor tissue quality has to do with fixative, okay? Remember what we discussed in the past, maybe in a case of maybe over-fixation or under-fixation. This is one of the factors that could lead to poor tissue quality, okay? Because what that is going to do, it will give you inconsistent staining. Remember, because of this poor tissue quality, depending on what the situation might be, whether due to autolysis or necrosis as a result of maybe even the tissue could be an old tissue, or as the case may be, or is not properly fixed, as the case, any factor could have contributed to that. What that is going to give is inconsistent staining, and that is not good. So poor tissue quality is one of the factors that could lead to poor IHC staining. So what can we do to be able to correct that? Number one, you might need to consider tissue handling. So the tissue has to be well handled. For example, ensure that there is a kind of quick and proper sample handling that could lead to quick fixation and all of that. That way it can be able to prevent the degradation of the biological activities or the biological component of this very tissue. So it has to be quick in the sample handling and fixation. That way we can be able to preserve the biological properties of the tissue. I hope that makes sense. So tissue handling is very, very important. It's important that we verify and validate, how the tissue should be handled. That way to maintain the standard when it comes to ensuring that we get good IHC staining. So also, where possible, we can also consider, avoiding using tissue that has been maybe wrongly stored, as the case may be, or maybe they are old, as the case may be. So avoid tissue that is wrongly stored, or avoid tissue that is old. That way we can be able to correct that. And that is where, like I mentioned on my previous video, we might consider, of course, using fresh samples where possible. So using fresh sample means we then need to avoid maybe tissue that has been inappropriately stored, okay, or maybe tissue that is old. Once we notice that the tissue has not been properly stored, or maybe that tissue is old, we might consider using fresh sample. I hope that makes sense, okay? So poor tissue quality is one of the factors that could lead to poor IHC staining. Why? Because of the possibility of autolysis and necrosis, and I've given you guys what that means. And of course, if the solution would then be to make sure that we handle the tissue sample very well in such a way that it can be quick, both in handling the sample, including the fixation and all of that. That way it can prevent the possible compromise of that very of biological properties of that very tissue. And of course, we can use fresh tissue where possible if we suspect it is old tissue, okay, old sample, or maybe if we suspect that the sample has not been properly stored. I hope that makes sense, okay? All right, thank you guys so much for listening. Please, can I ask you to like, share, subscribe, and comment. And I wish you all the best till I come back away again. Bye-bye.